Cholesterol. Cholesterol is this lipid molecule that has many carbons. As you can see, it has 27 carbons and it has an OH group attached to carbon number 3. And the more cholesterol you have in your membrane, then the membrane is going to get more fluid-like. So increasing the amount of cholesterol will increase the membrane fluidity. And the reason is because, as I mentioned last time or earlier, that these cholesterol molecules, they are very bulky in nature, or at least bulky in structure, and that is because they have 4 rings. Right? And these 4 rings will not allow these cholesterol molecules to rotate. And then as a result, when you have membrane that looks like that, and then these cholesterol molecules will just get stuck inside, and as they are going to be inside the membrane, they disrupt the bonding pattern of the different lipid molecules, and as a result, your membrane become, in a way, softer. Okay, so it's more liquidy, right? Liquid-like. Right? Cholesterol, it's important to higher organisms like animals and stuff because cholesterol is the precursor to many hormones. As you can see, we have the male sex hormone, the female sex hormone, and what you see is the general structure. Okay, the general 4-ring structure from cholesterol to the sex hormone type of molecule. And we'll talk about this next semester when we talk about biochemistry 2, is that your hormone molecules are actually derived from cholesterol. Okay, so this is one of the reasons why we don't encourage children that are too young eat too much things that have cholesterol, because it will stimulate premature puberty. This is one of the reasons, right? In addition to the high cholesterol, high calorie content. It triggers unwanted things, so you don't grow as tall as you're supposed to, that kind of stuff. In addition to hormones, you also got other functions that are going to be derived from molecules that are coming from cholesterol. And these are things like immunosuppressant, things that can help regulate blood pressure, and other immunosuppressants. So what you can see is that cholesterol is the precursor to many different molecules that have vital functions to controls, okay, to body controls, right? And these body controls are important for different purposes, right? So, this is just to get you introduced about this class of molecule and some examples of this function, but we will not talk about their biosynthesis until next semester. So in Biochem 2, we're going to go into detail about all the different enzymes that convert cholesterol to your hormones, as an example. So membranes, since we're talking about lipids, and of course one of the major functions for lipid molecules is to construct your membranes. Now, membranes, as we know them now, essentially they are a fluid. Essentially, it's saying that it's not static. Okay, things are moving all the time inside the membrane, and this is called the fluid mosaic model that was proposed in 1972. Essentially, what it says is that membrane is a fluid mixture of lipids and proteins, and fluid meaning that it can move around. So essentially, what you have is a bunch of proteins and a bunch of lipid molecules mixed together, but they don't stay at one place. Okay, they're constantly moving. Now, here we'll talk about 2 types of membrane proteins. We have the peripheral membrane protein, and the peripheral membrane protein are proteins that are associated with one side of the bilayer. It could be the inside or it could be the outside, whatever. It could be like inside or outside. But these things are just associated with the membrane. They are not in the membrane entirely. Okay, maybe a little bit of it touches the membrane, but not the entire protein. These proteins can be separated from the membrane without disrupting the bilayer. Okay, so you can simply wash them off, right? You don't have to destroy the bilayer to then separate these membranes. So they're only like associated, right? They're associated, may or may not be attached to the membrane. How do you associate with the membrane without being attached to the membrane? Association is like, let's say you have intermolecular bonding. Okay, so it's like attraction. For example, maybe you have on the membrane negative charge, and then your protein is positive charge. Then they can associate with each other. Attachment is more like the protein itself has a covalent bond with the membrane and it's attached there, or it's attached to the surface of the membrane through stronger interactions so that these membranes can sort of just sit there very tightly. Doesn't have to be covalent, it could be some other stronger interaction that just sits there that allow the protein to be more attached to the surface rather than associate with the surface. Association is more like reversible, you can on and off, but attachment is more talking about being on the membrane. So these things are the peripheral membrane proteins, right? So they're not in the lipid membrane. So that's actually the integral membrane proteins. So integral membrane proteins are proteins that are deeply embedded in the bilayer. Okay, so if you look at the picture here, that is essentially talking about this thing. You have the protein that's part of the membrane. So what kind of proteins would be a membrane protein? Transporters, right? Things that mediate transport from outside the cell to inside the cell. So those things are going to definitely be integral membrane proteins, right? They have to be in the membrane to do some stuff. So you got a bunch of different integral membrane proteins. Now, integral membrane proteins can only be extracted under conditions that disrupt the membrane structure. So if this protein is inside of the membrane, if you want to take this protein out, what you have to do is get rid of the interactions between the membrane and the protein. Okay, so you actually have to sort of dissolve the membrane and then separate the protein out. And this is not always easy. Okay, so you have peripheral and integral membrane proteins. So make sure you understand the difference. It's actually very simple. It's just, where is it? Is it on one side only, or is it in the membrane? Okay, if it's integral membrane protein, it's in the membrane. Now, so one of the things that we want to know is, how do you know it's a fluid model? It's actually quite simple. So what you can do is you can tag a protein. So membrane proteins and membrane components are constantly moving around and redistributing themselves. All you have to do is like tag a protein that's on one membrane, and then you then fuse the cells together, okay, fuse the membranes, and then what you should see is that these labeled proteins through time will get distributed across the cell. So this is maybe time 0, and then this is time whatever. And you can see how the different color proteins then can redistribute themselves, right? And that's a function of simple diffusion. It's going to rearrange themselves and then move randomly. So this movement we call lateral diffusion or lateral movement, meaning that they move in two-dimensional. Right? So essentially what you have is lipid membrane, and let's say you have a protein here, and your protein can move this way, can move that way, right? So it can move around. It can only move horizontally, because if you want to move vertically or flip-flop, we'll see that in just a minute, it's going to cost energy. But moving horizontal, no problem. Why? Because this surface is going to be hydrophobic, and this also hydrophobic. So hydrophobic and hydrophobic, they can move around, right? So membrane is not a static thing. It's always moving around. So then what kind of factors will affect membrane properties? So we already talked about the length of the hydrophobic tail will affect the property. Shorter the fatty acids, then the more fluidity the lipid membrane. Unsaturation of the hydrophobic tail. Like I said, the more alkene units you have in the tail, it's going to cause little kinks inside of the binding, and then as a result, the packing of the membrane will be lower. As a result, the membrane will be more fluid-like. Cool. Amount of cholesterol, we talked about that. The more cholesterol you have inside the membrane, it also serves as a way to disrupt the membrane packing, and then as a result, the membrane become more fluid-like, okay? More fluidity. Also, temperature. Temperature is very important. If it's cold, then the membrane is going to be more stiff. If it's hot, the membrane will be more fluidity. That makes sense. Just think of butter, right? Butter, if it's hot, it's liquid state. If it's cold, it's a solid state. Makes sense, okay? So temperature also affects your membrane packing. Now, you can go and make a synthetic membrane out of essentially detergents or surfactants, and usually if you want to make a synthetic membrane or just make whatever membrane, there are a couple things that you pay attention to. This thing called the transition temperature. Transition temperature is the temperature which membrane go from gel to semi-fluid liquid crystalline. Okay, so going from a more defined structure, amorphous type of molecule or amorphous type of state. Okay, so if it's in a gel, then you have very tightly packed, regular membrane structure. Now, if you heat it up, then essentially it becomes loose. And then where it becomes more of an amorphous type of structure, where you don't have a defined structure, right? You don't have a defined pattern for your lipid tails. So what you have is loosely packed head group, and tails will be disordered. And the membrane tend to be thinner. So when it's colder, membrane tend to be thicker because all of your molecules sort of aggregate. So you can think of it as a little sponge. If it's cold, they aggregate. If it's hot, it's like you're stretching it out. It has energy, right? Temperature is sort of a way to say you have a lot of energy. So you have energy, then the molecules are moving, vibrating. So essentially, the membrane gets sort of stretched out. Okay? It gets stretched out. If it's cold, the molecules, no vibration, okay, little vibration, so the molecules tend to pack together. So temperature affects the property of your membrane as well. Now, biological membranes at physiological condition is typically a semi-fluid crystalline state. So it's typically in this state. Okay, so at physiological condition, it's typically like that. That's why all your components, your proteins can move around, lipids can move around, right? It's because it's very loosely packed. So if we look at the composition of common biological membranes, these are the different kinds of membranes. On the left, you have myelin, erythrocytes, blah, 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 whatever. And then you have percent by weight, you got proteins, lipids, carbohydrates. Immediately, carbohydrates, you don't see that much. Why? Carbohydrates you don't find inside the membrane. You only find it outside of the membrane. Okay, either on the top or on the bottom of the membrane. Because carbohydrates are what? Okay, it's polar, so it's not going to be found inside of the membrane. It will be attached on the outside of the membrane. So carbohydrates you don't see too much. Now, the rest of the membranes are actually made out of proteins and lipid. Okay, like I said, back in high school, when we learn about lipid bilayers, we say, okay, your cell membrane is made out of lipids. But in reality, it depends on what kind of cell. If you have myelin, for example, it's about 80-20. 80% lipid and about 20% protein. But then, if you look at mitochondria, it's going to be 75 or 76% of protein and only 25% of lipid. So it depends on the type of cell. The amount of protein versus lipid will be different. And then you want to think about this. Okay, here's a thinking exercise for you guys to think about. Okay, what kind of membrane want to have a lot of proteins? And what kind of membranes want to have lower amount of proteins, like more lipid? Think about that. Okay, think about that. What kind of membrane want to have a lot of proteins, right? It's actually, you can think about a couple reasons why you would want that. Transporters, okay, yes, that's exactly right. If the membrane encloses a part, either a cell or an organelle, that requires a lot of transportation, okay? Maybe transporting molecules, transporting enzymes. If the membrane is responsible for an organelle, for example, mitochondria is an organelle, you have to actively transport pyruvate, you have to transport acetyl-CoA, that kind of stuff. You have to transport metabolites, transport energy. So you have to have cell and mitochondrial communication. If the cell needs to be able to communicate with the outside, then it will have a lot of proteins. Okay, so proteins are going to do what? Transporters, proteins can do reactions, right? So the more function you want with the cell or with this membrane, then it's going to have a lot of proteins. So things like myelin, for example, myelins are not going to be doing a lot of transports, okay? It's actually the shell of your nervous cell, okay? Your axon, and then you have the myelin shell. So the primary function for myelin is to protect the cell, right? It doesn't have to talk to outside too much. So think about what kind of cell you have, and then look at what kind of composition they have in terms of protein versus lipid. Makes sense, okay? Everything makes sense. So as we were talking about lateral, membranes in general, it's a fluid. It's dynamic. It's not static, it's dynamic. So we have lateral movement. That's very easy. Lateral movement is like this. So this is lateral movement. Lateral movement, super easy. You go from this guy to here. Very easy, right? Because top is hydrophilic. Bottom here is hydrophobic. So the properties are the same. So you can move and no problem. Okay? As long as you provide some energy in temperature, then it's going to move very fast. So it moves at about 1 micrometer per second. Is that fast? 1 micrometer per second? That's kind of slow, right? But whatever. For cell dynamic, it's pretty fast, okay? Now, if you want to move from top to bottom, like here, this is what we call a transverse movement, all right? Or you can say flip-flop. Okay? Essentially, you are going from the top leaf, okay? So we're going to call this is the top leaf, and this is the bottom leaf of the lipid bilayer. If you want to go from top to the bottom, all right? This is a very, very, very slow process. You do very, very slow. The half-life, the time constant is in days. This guy is in seconds. That guy is in days. So it takes a long time for it to move like that. Why? It's simple. If you want to move this guy to here, to the bottom, the hydrophilic has to pass through all this hydrophobic. That's not easy because hydrophilic passing through a layer of hydrophobic, nah, not going to happen. So if it's uncatalyzed, it will happen in days. So really just not going to happen. So there are going to be times that you want this to happen. Why? For example, let's say this is inside and that's outside. Remember like a while ago, like maybe last week, we saw something about lipid distribution, the bottom leaf and the top leaf of different cell membranes. And then we saw that, for example, for myelins, on top you have a bunch of different glycosphingolipids, right? And then on the bottom you have something else, right? So let's say you want to pump a lipid on the outside and you don't want it to be on the inside. What do? Right? That makes sense. You want the outside to have a specific type of lipid. Inside, you don't want so much. So what you have to do is use this enzyme called lipase. Lipase is a protein, well actually it's an enzyme. It can catalyze a reaction that uses energy of ATP, okay, and then it will change the conformation of this enzyme such that the enzyme will have a high energy, and then once it grabs your lipid, flip it on the other side, the conformation restores, and then it becomes the protein again until you have the next ATP coming in, and then it does that again. So you can do transverse movement, no problem, but you have to have an enzyme. You want to have an enzyme to do this process. If you don't have an enzyme, it's going to take days. With the enzyme, it takes seconds. So that's the purpose of an enzyme. It accelerates the rate of a process. Right? The process could be the reaction, it could be the movement, it could be whatever. So transverse movement. Uncatalyzed, super slow. If you want to catalyze it, if you want to make it faster, you have to use an enzyme to do it. So the two leaflet of the membrane usually differ in their lipid composition. So this is outer, this is inner. Lipid composition in the outer leaflet is shown in a green color, and the inner leaflet is a gold color. And if you look at it, you have three types of cells. You got the erythrocyte membrane, you have the liver plasma membrane, you have the platelet plasma membrane. So, different types of cells. And then here we look at 5 major lipids. You have the phosphatidylcholine, ethanolamine, serine, inositol. And then you have sphingomyelin. Now, you should roughly know what's the difference between these lipids. Choline and ethanolamine, they have what? Positive charge. Okay? So it's phosphatidyl, so it's a phosphate. So phosphate has a negative. Choline, ethanolamine has a positive charge. So these things are going to be sort of neutral in a way. Phosphatidylserine, if you go take a look at that. Okay, so by the way, PC, PE, PS, these three are sort of related. Serine, it has one additional negative charge on the outside, okay, because serine is the amino acid. The acid group is also on the outside, okay, whatever. All right, so, and PI, this guy is a sugar, right? Inositol is a sugar alcohol, okay? Sphingomyelin, you remember what sphingomyelin is, right? We just talked about it last Friday. Okay? It's that sphingosine thing with a phosphate thing on top. Okay? Sphingomyelin. So if you look at the distribution, what do you see? Okay? For example, erythrocytes and red plasma membrane, liver plasma membrane, you have a lot of PCs, okay, on the outside, and not a lot of PCs on the inside. And on the other hand, you have ethanolamine, PE, primarily on the inside and not so much on the outside. So knowing this, and then think back what we talked about in the previous slide about the lipase. So can you now see why you would need a lipase? Right? Because inside of the cell, your PC molecule is made inside the cell. Right? And then you want to get it on the outside. How do you get it on the outside? Right? You have to flip it onto the outside. Please understand that membranes are not symmetrical because the type of lipid that makes the membrane could be different between the top leaf and the bottom leaf. They could be different. And they're different for a reason, okay? Because what the function you need for outside of the cell and the function you need for inside the cell is different. Right? So of course, the lipid distribution could be different. So peripheral proteins, right? So like I said, peripheral proteins attach to membrane through association. These things are present on one leaflet essentially. Okay? So they only associate with one side of the membrane. Now, here's an example. This is the erythrocyte membrane, so your blood cell, and you have a bunch of different proteins that are peripheral membrane proteins, so the proteins that are associated to the membrane. And immediately I'm just going to tell you that these things are going to be related to the structure for this cell, maintaining the structure of the cell. So for erythrocytes, there are few proteins on the outside of the cell, and that is because erythrocytes' primary function is to carry what? oxygen. Okay, it's carry oxygen. It doesn't have to do a lot of stuff. It's not like an immune cell that has to attack stuff. You know, it doesn't do any of that. Okay? So its primary function, oxygen. And then oxygen works by diffusion. Okay, it's a gas, so you don't need special transporters or anything for that. It's diffused in and out. You can think of it as a plastic bag that has a lot of hemoglobin. So few protein on the outside, but on the inside, you got a lot of peripheral protein. It has a 2D network of things that are made out of actin. This thing called band 4.1. This thing called spectrins. Is it important you know what these things are? No, okay? I don't care if you know exactly what these things are used for. No, I want you to know the general property of what's on the inside of your erythrocytes. Take a look at this picture. What do you see? You see a bunch of proteins here, and they're attached to your actin filaments. These things are the spectrin tetramers. Okay, what does that look like? The guy said maybe it's like a little road for things to move. Okay, maybe, I give you that. Anything else? Okay, so for me, it looks like a net. Okay, it's like a racket or a badminton racket. It looks like a net. Okay, so it's got a mesh of proteins that's associated to the membrane. So what does it do? It sort of maintains the shape of this particular cell. So you got all of these proteins there, and they associate with the membrane. They don't go into the membrane too much. You do have channels that go into the membrane, but you know, here, these guys, they just associate with the membrane. Okay, and essentially, what these proteins do is they sort of push against the membrane and then provide a structural support. Okay, so this structural network of these proteins provide an excellent support for the erythrocyte to maintain its shape. Why is that important? Because your blood cells have to move through very different environments, right? It could be in the artery, it could be in the veins, it could be in small capillaries. So it has to move at different places. So it has to be very flexible, right? So it's one of the applications of your peripheral membrane protein, if it's a structural support. Integral membrane protein is more exciting. That is because integral membrane protein do a lot, and there are a lot of proteins that are integral membrane protein, especially transporters, like we talked about, signaling molecules. How do you get signals from outside the cell to inside the cell? Right? How do you signal? How do you know, like, for example, your blood glucose is high, right? You're going to have a signaling molecule that's going to circulate in your body to tell you, okay, you just ate. And then your cell will adapt one metabolic state. Right? And then when you're hungry, your cell will become another metabolic state. But how do your cell know that? It knows it by having signals, okay? And then signals have to bind to some kind of receptor that's on the membrane. And then the receptor changes the shape, and then it triggers some inflammation inside the cell. Have you guys heard about signal transduction back in general biology? This is for that purpose as well, right? Now, so what you see is that integral membrane proteins will contain hydrophobic residues that promote association with the hydrophobic tails of the membrane. So what you have is here, if this is the membrane, on the side of that protein, you're going to expect to find amino acids that are hydrophobic. So the residues on the side here will be hydrophobic. And regardless of the size of the protein, that is going to be true. And here you got some very interesting shapes. You got porins, which are molecular transporters. You have bacterial rhodopsin. This is super exciting. It absorbs light and it creates proton gradient, et cetera, et cetera. Or you have a crenal fumarate reductase, which is complex 2. Right? So you can see how you have different size integral membrane proteins. Okay? Some could be really, really big. Some could be really, really small. Now, transmembrane proteins usually have transmembrane domains. So, unfortunately, for introductory biochemistry class, we don't have the time to go into the different domains of proteins and enzymes, but I want you guys to just understand this general description. Okay? Proteins have domains. what are domains? Domains are like a portion of the enzyme that has a specific function. Okay, so that's like a domain. It's a big portion or a chunk of the protein that has a specific shape or a specific function. We usually just call that domain. So this word, you'll find this word a lot if you're dealing with huge proteins. So transmembrane proteins, they typically will have domain structures that are going to be hydrophobic. So these hydrophobic, or at least these transmembrane domains, right, are typically derived from alpha helices or beta sheets, okay, very regular patterns that have a lot of hydrophobic residues on them so that they can interact with the lipid tail of your membrane lipid. So, for example, if you take a look at bacterial rhodopsin, this enzyme has a vitamin A inside it. It absorbs light. After it absorbs light, it can pump a proton into the other side of the membrane, and as the proton comes back down, you can generate ATP. So bacterial rhodopsin is a very useful enzyme for organisms that can absorb light. And what you can see is that for bacterial rhodopsin, the transmembrane domains are made out of alpha helices. On the other side, you have an OMPF porine. Porine is a transporter. What you see is that both of them are transmembrane proteins, but porine, the structure is made out of a lot of beta sheets. Okay, so you have beta strands that essentially make you beta sheets. Now, if I want to ask you, what do you think would be the amino acid pattern from this strand? What do you think? So facing inside here, you have a channel. So that probably has to be hydrophilic. Outside, which is touching the membrane, has to be hydrophobic. Okay, so what do you know about beta sheets? That they alternate, right? So every other R group sort of faces one side and the other side, right? So this R group points up, that R group will point down. Okay, so then you'll have alternating R groups that differ in polarity, right? So one side will be polar and the other side will be nonpolar. Okay, so this is something that you can probably expect. I wouldn't say that is true for all transporters, but at least you can expect some kind of a pattern. So if you take a look at the integral membrane proteins and their amino acid sequence, here given is the sequence and the postulated structure of glycophorin A. This is a protein used to carry sugars, in particular sialic acid on the red blood cells. Now, if you look at this particular protein, and what you see is that you have the external, outside the cell, cytoplasm is inside the cell, and you have this part that is the transmembrane domain. What you see is that you have a single transmembrane helix that is highly hydrophobic. So here you have a sort of a streak of non-polar residues. Isoleucine, threonine, leucine, isoleucine, isoleucine, phenylalanine, glycine, valine, blah, blah, blah. Right, so these things, this string of non-polar or hydrophobic residues is predicted to be inside of the membrane. And then on the outside of the membrane, you have primarily hydrophilic residues that will bind whatever substrate that they bind. So what you see is that the cytosolic C terminal is quite hydrophilic. So here, I guess, quite hydrophilic. And then on the N terminus, you have a bunch of sugar that are attached to it. So what do you see? This is a very typical membrane protein that you can find on the membrane of your cell. Outside, so this is outside, external, you have a bunch of hydrophilic residues, and these hydrophilic residues will be attached to sugars. Okay, they have sugar modifications. Inside, usually not so many sugars, but still hydrophilic. Across the membrane, you have hydrophobic residues. So using that kind of idea, you can actually construct some kind of a map that looks at the relative polarity of the residue and in the sequence of your protein. So for bacterial rhodopsin, you have 7 helices that span the membrane. So you're going to have 7 helices that are going through the membrane. So it's go into the membrane, out, in, out. Okay, so you have 7 helices that's inside of the cell membrane. And if you look at the hydrophobicity plot, meaning from sequence residue number 1 to residue number 240, and then you list based on their hydrophobicity. How do you list them? Like leucine, very hydrophobic, phenylalanine, very hydrophobic, aspartate, not hydrophobic. And then you plot it. What you see is you're going to have alternating chunks that's sort of hydrophobic. So you start the protein at the very beginning, both the C-terminal and the N-terminal, they have to be hydrophilic, usually. Why? Because they're usually exposed to the liquid media, either outside or inside. So usually the N-terminus and the C-terminus, they're going to be hydrophilic, because usually they're on the outside. Very soon, you start entering the first helix, and then comes back out. So you see how there are residues on the outside, residues on the inside. And then you got some residues here, and then you have another span of your hydrophobic islands, like a lot of hydrophobic residues. Because if the helix is inside of the lipid membrane, you will not have alternating hydrophobicity. You will not have, that every 4 amino acid you have a hydrophobic. No, you don't have that, right? Because if it's inside, all amino acids will be hydrophobic, right? Because your protein is going to touch all sides of the membrane, okay? Right, because your protein is inside, and then surrounding the protein, you have lipid. Now, you will still find a little bit of maybe hydrophilic residues because they have to do some interaction between the helices. But other than that, you see a lot of hydrophobic residues. Right? So if you plot this, and then you see hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic. Right? So each of these chunks that tells you where the transmembrane domains are. Okay? So knowing this, you can program your software to help you recognize transmembrane domains. Right? Okay, so the more proteins like this we have, then you can use bioinformatic tools to then take whatever unknown sequence you have, analyze it, and then as simple as you can do a hydrophobicity plot, and then you can see alternating patterns of hydrophobic islands, like hydrophobic chunks, hydrophilic, hydrophobic chunks, hydrophilic, like that. You can sort of get an idea what this protein might have. Okay? In this case, it might have a transmembrane domain. So integral membrane protein, it's just how it is. So then the question is, okay, do you ever think about, membrane protein, it's going to be hydrophobic, but then ribosome, it's inside the cell, okay, and you are using ribosome to make proteins, okay, and ribosome is inside the cell is aqueous, okay. So then, as you're translating this messenger RNA into membrane protein, would you think that immediately all your hydrophobic parts will just like group together and then you will produce an insoluble protein? That makes sense, right? Right? So it turns out there are going to be accessory proteins that you have to use in order to get your protein into the membrane. So this is like way oversimplified scheme, but for our purpose, we don't have to get any deeper than this. If you guys are interested in this, you can look it up. So generally speaking, you have a ribosome, and ribosome is going to make you your polypeptide. Now, what happens is that you will have ribosome on top, and so you have some kind of a mRNA, and it's doing translation. So as it's doing translation, the polypeptide comes out, and then what happens is that the ribosome will get close to the membrane, and you will spit out this polypeptide chain, and this chain will go into a hole. It will actually go into a channel that's inside of this pre-existing protein complex on the membrane. Okay? And then you're going to ask me, like, where do you get this guy from? You curious about that? Not curious? It's okay. I'm not going to tell you. I'm just kidding. All right. Like when your cells divide, right? It already has a membrane, right? So it already has whatever that's there. Okay? And then you're going to ask me, then where does the first membrane come from? I don't know. I don't know where the first membrane comes from. Evolution? Okay? But whatever. Initially, on the membrane, you have some accessory proteins that's there to help you fold your incoming protein. Makes sense. Because otherwise, your ribosome make that protein, and the protein is like, oh, I'm hydrophobic, and I'm going to collapse together, and then that's it. You're done. Okay? Your protein is dead. Dead protein. No good. So what happens is that your ribosome spits out a protein that looks kind of ugly, but it spits out a protein. And then this protein goes into this protein complex, and the protein complex will sort of help it fold. And then as it matures into that whatever shape that it's supposed to have, it sort of slide it on the right, and then you're ready for the insertion of the next helix or beta sheet, whatever. And then you repeat on doing this until all of the protein ends up here. And this is the regional structure, and then together they form into the tertiary structure. So this is like a side view. So if you look on a top view, what do you have? Okay, you have this thing called the translocon. So initially this guy is plugged, and then you have this hole here that you can start doing your stuff, and your ribosome is going to spit out your polypeptide into this hole. It's going to, in a way, fall slightly into the hydrophobic core right here. So the core here is hydrophobic, and then it's going to fall, and after it's done, it's going to get kicked out like this. It gets kicked out, and then this ring closes again, and then you can continue to keep on inserting polypeptides until the entire polypeptide sequence ends up on the membrane. Now, this bottom right picture here, I want to just show you how membrane proteins, they actually have directions. Right? You're like, whoa, if it's a receptor, you want the receptor to be on the right side, right? If the receptor is upside down, that's kind of stupid. Okay, so your membrane proteins have directions. Okay, whichever side is supposed to be on the outside, it better be on the outside. And one of the ways that you can use to do that is, for example, you have charges. This is more or less E. coli membrane. Okay, E. coli, we have this thing called periplasm and this thing called cytoplasm. Periplasm is where the proton gradient is, so it's positively charged. Cytoplasm is primarily phosphatidylethylamine, that kind of stuff. Okay, so it's just membrane, right? So it's going to be negatively charged. Okay, you have a protein that is going to be transmembrane. Okay, the wild type protein is going to be facing the N-terminus and C-terminus on the outside, and the middle is on the inside. Okay, and that is because you have positive charge on the inside. Okay, so it's sort of oriented itself in that way. Now, all of a sudden, you're like, I don't like that. I want to change the protein structure a little bit. And then you put the positive charge onto the N-terminus. And then what you see is that you actually have a flipped structure. So the local environment on the membrane also helps to sort of differentiate which end is the proper end. This is the last slide of this lecture, and that is membrane rafts. Okay, raft 有没有人知道什么？你拿几根木头叠在一起，一个平板，然后你可以站在上面，有没有？那东西叫 raft. Then what is like membrane raft? It's like, what? Okay, membrane raft or lipid raft. Turns out these membrane rafts are essentially like little islands inside of the fluid membrane. Okay, we just said that membrane is a fluid, so it's like moving around. So you can think of membrane as the open sea, I guess, okay? And then you can have rafts. So rafts are these little chunks of stuff that aggregate, and then together they move simultaneously. So membrane rafts are rich in cholesterol, sphingolipids, and GPI-linked protein. Right? You're like, well, what is GPI? GPI is that glycosyl phosphatidylinositol. So this is a lipid. It's a lipid protein, right? Whatever. Membrane rafts, essentially it's rich in cholesterol. Okay? So you form these little islands, and these little islands will move together in the membrane. Once you form these little islands in the membrane, the bilayer is thicker. Okay? It actually becomes thicker. So let's see, where is the lipid raft? Okay, so this is a lipid raft. And what you see is that it has a lot of these GPI anchor protein, and it's going to have a lot of cholesterol. Cholesterol is this little thingy right here. These little thingies. All right, so they sort of aggregate together, and they form this nanometer size dynamic raft domains. So together, then they move. And rafts can associate to form larger structures like platforms. Okay, so actually, I guess this is actually a platform rather than a raft. Raft would be something like this. You have a lot of cholesterols right here together. Okay, so that would be like a little raft. Rafts together, you form a platform. And what we see is that certain proteins interact preferentially with the rafts, and some other proteins do not. And that just tells you that, you know, these things exist. Okay, I don't know what it's for. I'm just kidding. I know what it's for. But if you're interested, we can talk about that later. All right.